called MEC, versus temozolomide, which is an oral chemotherapeutic agent for patients with advanced uveal melanoma. So just in the way of background, as you heard, uveal melanoma is a rare type of melanoma that arises from the eye. It is an orphan disease, which means it's quite rare. There are less than 2,500 cases diagnosed each year in the United States. And although this is a disease that arises from melanocytes, just like cutaneous melanoma does, this is distinct biologically from cutaneous melanoma. And unlike the case for cutaneous melanoma, as you've heard, for which there are effective therapies, there are no effective systemic therapies for metastatic uveal melanoma. Current standard of care for these patients, therefore, is clinical trial participation. Now, in 2008, it was found that there are frequent mutations in genes called GNAQ and GNA11, and it was found that these activating mutations activate a pathway called the MAP kinase pathway. And preclinically, we demonstrated that inhibition of that protein called MEC, which shuts down that pathway, leads to cell death in uveal melanoma. And so therefore, we sought to assess the efficacy of selumetinib, which is this MEC inhibitor, in patients with metastatic uveal melanoma. To do that, we conducted a randomized trial where patients with metastatic uveal melanoma who have not received prior chemotherapy with temozolomide or DTIC were randomized on a 1-to-1 ratio to either chemotherapy with temozolomide or selumetinib. The selumetinib was administered orally twice daily on a continual basis. Patients who were randomized to temozolomide were allowed to cross over to selumetinib at the time of radiographic progression. Now, at baseline, before randomization, all patients had tumor tested for mutations in GNAQ and GNA11, and so randomization was stratified by mutation status, extent of disease, as well as number of prior therapies. The primary endpoint of this trial is progression-free survival. Secondary endpoints include response rate as well as overall survival, and patients had radiographic assessments by RECIST 1.1 performed at weeks 4, 8, and then every 8 weeks subsequently, and we treated patients until progression, intolerable toxicity, or withdrawal of consent. In terms of randomization, the arms were well balanced in terms of age, gender, performance status, stage, and number of prior therapies. Greater than 90% of patients in both arms had advanced disease with liver involvement, which is the most common site of metastasis in uveal melanoma, and greater than 50% of patients in both arms had an elevated LDH, which in cutaneous disease is a poor prognostic marker. Again, all patients had tumor screening for GNAQ and GNA11 mutation status, and you can see that 37% of patients treated with selumetinib had a GNAQ mutation, 44% had a GNA11 mutation, 19% were wild type, and the numbers were fairly similar on the temozolomide arm. What we observed is that the response patterns differed between arms. What's shown here is the waterfall plot for patients randomized to temozolomide. There are 46 patients evaluable for response. And what you can see here is just a small minority of patients had any sort of radiographic regression, consisting of just 11% of patients, and no patient had a major response in terms of RECIST. In contrast, this is the waterfall plot for selumetinib. You can see here that 50% of patients achieved major tumor regression, and 15%, or 7 of the patients, actually had a RECIST response. And this was quite remarkable because radiographic shrinkage in this disease is quite uncommon. And in fact, if you look at a series of previously published phase 2 trials, response rates are less than 5%, whether you're looking at various combination chemotherapy agents, immunotherapy, or targeted agents. The primary endpoint was progression-free survival, and we did demonstrate an improvement in progression-free survival. With temozolomide, the median was 7 weeks versus 15.9 weeks for selumetinib. This was highly statistically significant with the hazard ratio, which I'm sorry is not on the slide, is 0.46. So that's a 54% reduction in the risk of progression or death, highly significant in terms of p-value. And despite the crossover design where more than 80% of patients initially randomized to chemotherapy crossed over to selumetinib, there was a trend towards improved survival from 9.4 months to 10.8 months. This was not statistically significant. So in conclusion, this study is the first to ever demonstrate efficacy of any systemic therapy in patients with metastatic uveal melanoma. MEK inhibition with selumetinib results in a progression-free survival double that achieved with chemotherapy, improving median PFS from 7 weeks to 15.9 weeks. Tumor shrinkage is achieved in a significant proportion of patients treated with selumetinib, and selumetinib could be considered a new standard for patients with uveal melanoma and provides a platform for the development of new combinatorial therapeutic approaches. Thank you.